Manage the budgets can be quite tricky, especially when you don't have much. Imagine your budget is tight this month and you've critically planned it to the last rand, and then boom, 5.9 billion of it vanishes. Well, actually, I'm talking about South Africa's health budget, and believe me, it's tight. The 2012-2013 health budget was that of 121 billion. And an anti-retroviral tender for the same period was that of 5.9 billion, where active pharmaceutical ingredients accounted for about 60% of the cost.
Therefore, it is of no surprise that South Africa is considering producing its own ARV APIs in an endeavor to save the government of money and secure the ARV supply.
So, as pharmaceutical researchers, we have a significant role to play. Lowering manufacturing costs may be done by simplifying manufacturing processes.
Research highlights that registered drugs in our market, such as ARVs, have a low market value due to their poor physicochemical properties. More so, current fixed-dose combinations on the market require multiple excipients, leading to large undesirable dosage forms. Therefore, finding novel approaches of combining two APIs has always been an area of research interest.
However, finding the most cost-effective manufacturing method of combining two drugs, now that's a challenge. But drug-drug co-crystallization offers an alternative approach with the possibility of fewer excipients needed.
A drug-drug co-crystal is defined as a multimolecular component bonded non-covalently, where one of the components is a neutral API and the second component is a coformer, which may be neutral or ionic in nature.
Please note, a definition that goes against the normal grain. We challenged previous literature that outlined a clear distinction between a salt and a co-crystal, and we did so in accordance with recent literature, where polymorph co-crystals such as peroxycam have been discovered in both the neutral and salt form, where a neutral API weakly associated with the neutral part of an ionic coformer to form a salt co-crystal. Therefore, we aim to explore the possibility of salt co-crystallization using sodium valproate for its neuroprotective attributes with anti-retroviral therapy.
So, in a nutshell, the main aims and objectives of this study was to investigate the possible formation of a drug-drug co-crystal for clinically relevant ARV therapy. Secondly, it was to investigate the possible formation of a salt co-crystal using sodium valproate. And lastly, to identify the most favorable drug-drug co-crystallization preparation method.
Please note the methods and analyticals used in this study.
So what exactly did we find in our study? Well, we have identified that solvent drop grinding is the most favorable co-crystallization preparation technique. This was because solvent drop grinding was the only method that produced testable solid or semi-solid material for all of our combinations. Slow evaporation only produced testable material for two combinations, while sublimation produced no testable material.
So let's dig deeper into our actual findings. From this point forward, we'll only elaborate on the most important findings. So DSE analysis is very much the same as playing spot the difference. We look at our new combination curve and we try to spot the difference with melting or degradation points. These may be in the form of a shift, a broadening, or disappearance in curve segments.
Exploring the formation of a possible salt co-crystal using sodium valproate in our first three combinations proved not to be possible. This was because in combination 1 curve, we saw the presence of all reference peaks with nevirapin's reference peak seeing shift. However, this was not due to polymorphism because it was outside of the ranges found in literature. Further literature attributed this to be a baseline shift, meaning that the near combination caused the shift, leaving only a physical combination behind.
Combination 2 produced a gel residue following dry grinding and solvent drop grinding methods. Therefore, powder X-ray diffraction was a better analytical as it could detect if any solid material was present. The definition of a co-crystallization requires a solid to be formed. Again, any differences from the original baseline patterns indicate possible interactions. Both graphs depict a straight line with no diffracting patterns. Therefore, it can be concluded that no solid material was present and that it was in fact a wet physical mixture.
So in combination 3, we see a new exothermic curve which is attributed to solvent loss, while the new endothermic range was confirmed to be a shift in the degradation of zidovudine. Literature attributed this shift to the thermodynamic properties of zidovudine on exposure to increased temperatures. This may be the case with our research, as heat exposure was part of our analytical procedures. Now let's have a look at our neutral combination.
So combination 4 showed a new peak, which was thought to be a shift in the AZT reference peak. However, this peak was not seen in our other analyticals. It may be attributed to the thermolabile properties of AZT. The sample showed a degradation point similar to that of AZT, but before the melting point of MEP. DSC clearly shows the absence of MEP. These findings suggest that only AZT or AZT with almost undetectable amounts of MEP was present in the sample. Therefore, a physical mixture was concluded.
So finally, the crux of our findings, the formation of a drug-drug co-crystal of an ARV combination. So remember the spot the difference analogy? Well, here's where it could not be missed. The DSC curve shows a clear absence of both reference peaks. More so, new low-intensity peak occurring at 71 degrees corresponding to methanol solvent loss. The second new peak at 108 corresponded with both TGA and HSM analysis and was therefore attributed to a chemical interaction between zidovudine and efavirenz. The almost complete broadening you see in the DSC curve, together with the low-intensity broad peak seen in powder X-ray, indicates an amorphous sample. The additional new high-intensity peaks in powder X-ray were indicative of a crystalline substance.
In FDR analysis, we saw a shift in the functional groups compared to the reference drugs, showing the formation of a new non-covalent bond. The significant amine stretch of efavirenz and carbonyl stretch of AZT represents a weak interaction. Per definition, co-crystallization involves weak interactions. The bonding groups mentioned previously are shown through literature to form a carbamate bond, which are shown to be weaker than lone amine bonds. Therefore, the study established that AZT forms a co-amorphous co-crystal with efavirenz by means of solvent drop grinding with ethanol.
This research study has enabled us to gain understanding in a relatively unexplored field of drug-drug co-crystallization. So what exactly have we accomplished with the active pharmaceuticals at our disposal? Firstly, we have identified solvent drop grinding as the most favorable method for drug-drug co-crystallization. This was confirmed in literature.
Secondly, we explored the formation of salt co-crystals using sodium valproate. No salt co-crystals were, however, obtained and proved to be physical mixtures, as no significant interactions could be confirmed on analysis. During the research process, we have come to understand that failure is sometimes part of the learning process. However, we would like to think that we haven't really failed, we just found 10,000 ways not to do it.
We then investigated the possible formation of a drug-drug co-crystal that did not include a salt. Combination 4 proved to be a physical mixture, while combination 5 showed positive results. More specifically, it turned out to be a co-amorphous co-crystal. So what exactly is a co-amorphous co-crystal? It's when crystallization happens between one amorphous substance and one crystalline substance, and they come together to form a co-crystal. The most likely site for this interaction was between the amide of efavirenz and the carbonyl of zidovudine. This brings us to the first limitation. The exact non-covalent interaction could not be visualized with the analyticals used in our study. Further research could be focused on eliciting its three-dimensional co-crystalline structure.
Efavirenz and zidovudine, together with lamivudine, form part of the first-line anti-retroviral therapy in South Africa. Co-crystal forming abilities between zidovudine and lamivudine have already been explored by other researchers, and the co-crystal found in our study between zidovudine and efavirenz should also be further explored for its potential in novel drug formulation. The success of co-crystals in pharmaceutical manufacturing is tied to their unique chemical composition, crystalline structure, and pharmacophysical properties. What is clear is that co-crystals present opportunities, both commercially and in the form of patents. And the way to take up this opportunity is to engage in further research. We hope that you've gained insight into the concept of co-crystallization and recognize that drug-drug co-crystals have their rightful place as combination therapy in the future of pharmaceutical drug development.